Now the next disorder is sickle cell anemia. It is not X-linked because its gene controlling it is present on the 11th chromosome. So it is an autosomal Mendelian disorder. And remember, this is also recessive. So all the disorders you have studied so far are all recessive disorders. And what happened in the case of sickle cell anemia is, we all know hemoglobin carries 2 alpha chains and 2 beta chain. Now what exactly happened with the beta chain of the sickle cell anemia person is that the hemoglobin will become modified. Why there is a modification? We all know primary structure of protein is made up of a fixed number of amino acids and fixed sequence of amino acid. Now what exactly happened in the sickle cell anemia is the beta chain of the hemoglobin has a fault or a wrong amino acid attached at the 6th position. So what exactly happened, why exactly it happened, we will be understanding in detail in the next chapter, because again mutation topic is this same mutation topic. So 6th position in a normal hemoglobin, beta chain carries glutamic acid, but because of a mutation, glutamic acid is now replaced by valine. So one amino acid change, and this will lead to a disease called as sickle cell anemia. This shows how drastic can be the effect of even a small scale mutation. Just one amino acid is changed and the person has a disease called as sickle cell anemia. Now that's why when we write symbols, we write for pedigree analysis HBS for a person whose hemoglobin is sickle cell anemia type. This is a person who will die because of severe anemia. This is normal, this is abnormal. Remember, HbA is normal. HbS will be heterozygous, so this individual is going to survive. Okay, now there are benefits also of heterozygous situation, especially in the countries or continents like Africa. A lot of malaria epidemic is there. So malarial parasite is not able to survive in a sickle cell anemia type RBCs. That's why such individual which are heterozygous, they are having an upper hand in the course of evolution, because malarial parasite is not able to survive inside such sickle shape RBC. Now why this shape change occur? This shape change occur only when there is less amount of oxygen carried. So whenever there is oxygen deficiency, the RBC shape is going to change. It changes because hemoglobin undergoes change in its structure, 3-dimensional structure. So accordingly RBC shape will change, and such RBCs are not able to carry oxygen properly. So this will cause oxygen deficiency. And finally such deformed RBC, okay, why is it called sickle cell? One point you have learned. Now these RBCs will go into spleen and destroy them, and spleen destroys them at a greater rate because many of them are bad. So suddenly there will be deficiency of hemoglobin, or you can say anemia. That's why it is called as sickle cell anemia or hemolytic anemia, because breakdown of RBC has caused this anemia. So sickle shape of the RBC and hemolytic anemia are the two main points of sickle cell anemia. Let's understand this from the NCERT. And this is one of the favorite one to come in the board exam. Sickle cell and thalassemia, one more is coming on the next page. It is an autosome linked recessive trait that can be transmitted from the parents to the offsprings when both parents are carriers. So HbA, HbS, one parent, HbA, HbS, second parent. So when such parents undergo marriage, now the next generation may have the chances of HbS, or such person will not survive. The disease is controlled by a single pair of allele, HbA, HbS. Out of the 3 possible genotypes, only these individuals will show disease phenotype. Which one? HbS and HbS. Heterozygous individual appear apparently unaffected, but they are carrier of disease, as there is 50% probability of passing the mutant gene to the progeny, because 50% gametes will have HBA, 50% will have HBS. So 50% probability of transmission to the next generation, thus exhibiting sickle cell trait. Defect is caused by substitution mutation, in which glutamic acid is replaced by valine at the 6th position of the beta chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution, so it's a point mutation. Remember, few days back we learned what is point mutation. Single base or single nucleotide change is called as point mutation, and the opposite of this was gross mutation. So substitution of amino acid is due to the single base substitution of the 6th codon of the beta gene from GAG to GUG. Actually, there is a mistake here. This is not for gene, this is for mRNA. The mRNA which carries information is GAG, which is now replaced by GUG, because U is not present in genes, U is present in RNA. The mutant hemoglobin molecule undergoes polymerization. So this is the answer why the shape changes. It undergoes polymerization under the low oxygen tension, causing the change in the shape of the RBC from biconcave disc shape to elongated sickle-like structure. So NCERT has given this explanation here. The mRNA carries GAG, so GAG, and the mRNA now carries GUG. So GAG, basically this was the problem here, that a single base has changed, and this has caused a change in the mRNA. This has caused change in the mRNA, and finally change in the amino acid, and this is called as sickle cell anemia.